The next section is a discussion on enzymes. Enzymes are those molecules that are involved in all these metabolic reactions that we've been talking about. And when we talk about an enzyme, let's first consider what is an enzyme. So enzymes have very important jobs in our cells. The first thing they're a catalyst. So a catalyst is something that's involved in a reaction, but it's not used up in that reaction. So it can be reused over and over again. The other thing about enzymes is they are very specific for what reaction they run, because they actually have to bind the substrate or the reactant, the part of the reaction that is first needed in order to run the chemical reaction. And what are enzymes made of? Well, enzymes are actually proteins. So our proteins are coded for by our genes. Remember, they get copied into a messenger RNA, and then the protein is constructed using the ribosome in the cell. And so our enzymes, because they have to go through all this process, it makes sense that they are reusable. Here's some example of enzymes, and just listen and see if you can pick up what's going on. RNA polymerase makes RNA. DNA polymerase makes DNA. Lactase breaks down lactose, a milk sugar. Lipase breaks down lipids or fats. So if you realize there's something similar in all of these words, and that is the end of them, that they all end, most enzymes end in an ASE. So polymerase, lactase, lipase. Now if you actually look at these words, it tells you what they work on. Lactose. Lactose is a milk sugar. Remember, that's a disaccharide. Here is an example of an enzyme called sucrase. And sucrase breaks down sucrose. We learned about sucrose in our molecules class. Sucrose is a disaccharide made up of glucose and fructose. And so when water is used, so this process is a hydrolysis reaction. Hydro, when we break down something with water, and in the process we actually break it down and make glucose and fructose. So the water gets used to recreate that bond that was split when you broke it down. So we add in water in order to break down. That word is lysis. What about lactose intolerant people? If you're lactose intolerant, you cannot break down the milk sugar lactose, and you have all sorts of funny tummy problems when that does happen. But because this enzyme works in your stomach, you can actually take a supplement and get it right to where it needs to be. So most of our proteins would get digested like a steak dinner, but an enzyme that has to operate in your stomach, you can just take it orally and then you can break down your milk sugar and enjoy. What enzymes really do is they actually overcome something called the activation energy barrier. So anytime you have a chemical reaction between molecules, we either breaking bonds or we're forming new bonds. And in order to get this reaction going, like a jump start to help it happen, it's called the free energy of activation. Now, if we didn't have this activation energy, then it would be just mass chaos in our cells, because reactions would be happening all over the time. So it's a way to prevent spontaneous breakdown in molecules or spontaneous buildup of molecules. This activation energy is often in the form of thermal energy that comes from the surroundings and helps the reaction to start. Here's an example of activation energy, a visual of it. You can think of this activation energy as a barrier. And so what's happening is the reactants, every now and again, they bump into, the bonds are strained and they become products. If you have an enzyme, you're basically lowering that barrier so the reaction can happen much quicker. At no time does the free energy of this reaction happen in any way. The enzymes help with that energy of activation. So here's actually showing a schematic of it. Here's your free energy of your reactants and your free energy of your products. In order for this reaction to happen, we need to get these molecules into a transition state. And you can see here that the bonds between them are actually being strained. And then once the reaction starts to proceed, we end up with our products. And you can see A is now bound with C, and B is bound with D. And overall, this is a spontaneous reaction because delta G is less than 0. So enzymes help reactions by lowering the activation energy. They do not affect the change in free energy, but they help reactions happen that eventually would occur because the right enzyme, the right molecule would be turned into its products. Without an enzyme, this is a very, very slow process. So this next graph actually shows you a perfect example of the course of this reaction without an enzyme. You can see the activation energy in order for this reaction to happen is much higher than when you have an enzyme. The reaction energy is decreased, and your products are still the same that would have been made with or without an enzyme. So enzymes just help reactions go to completion. When we talk about enzymes, how do they actually work? So the enzymes have because they're proteins, they have a very important portion on the enzyme, and we call this the active site. And the active site is what's going to bind our substrate. And when our substrate binds to the active site, we get what is called the induced fit. You can almost see that protein changing shape around its substrate, straining those bonds and allowing that reaction to happen. We call this the enzyme substrate complex. So what is actually happening on that active site? Well, there's going to be an interaction between the amino acids. These are some amino acids inside that pocket on the protein, the enzyme side. And you can see when the substrate comes in, it's going to interact with whatever proteins whatever amino acids make up that active site. And so if I had hydroxyl groups, we might get some hydrogen bonding. We might get a disulfide bridge. Here we can just have some ionic bonding forming, some hydrogen bonding, more hydrogen bonding. And so there's always going to be an interaction between that substrate on its active site. And so the active site orientates the substrates correctly. It strains the substrate bonds. It's basically providing a favorable microenvironment for that reaction to happen. Some enzymes even covalently bond to their substrate. Here you could see some disulfide bridges. And so all of this is going to affect that reaction. And so you can see if we happen to remove a very important amino acid out of the active site, then maybe it would not bind its substrate properly, and this reaction would not be able to proceed. So our proteins, their structure is so important, and different parts of proteins are more important depending on what that protein does. So definitely amino acids that are involved in active site formation, inside this pocket formation, are going to be more crucial than, say, an amino acid that's formed way out here on the outside. But amino acids, you can see the amino acids in this area here that are interacting with our substrate. Those are going to be crucial for the function of the enzyme. So let's look at this process showing two substrates binding to an enzyme, held in a favorable environment by some weak interactions. The activation energy is lowered, and then the substrates are converted into product. The product is released, and the enzyme is now available to run the whole reaction again. So recycling, reusing that same enzyme over and over again in order to run a reaction. So let's look at some things that could actually affect an enzyme. The most important thing is thinking about temperature. because temperature affects the structure of a protein. If the temperature gets too high, the protein will denature, and when a protein denatures, it's no longer in its nice three-dimensional form, and so its active site will be affected. So this is a graph showing optimal temperature for two enzymes. The first enzyme loves to operate around 37 degrees Celsius. That is the normal operation of human enzymes. Here's an example of a heat-tolerant bacteria, which can grow up to temperatures of 95 degrees, and so its enzymes like to work around 77 degrees. If we took a human enzyme and put it at 77 degrees, that enzyme would no longer function. So temperature is very important for the operation of an enzyme. We'll be doing a lab on enzymes, and some of you will investigate the effects of temperature on a specific enzyme. And it's also going to depend on where that enzyme was isolated from. So if we're using an enzyme that came from a potato that normally grows in the ground, it's going to have a different optimal temperature or a different range of temperatures to, say, an enzyme that works in the human body at 37 degrees. Another thing that can affect enzymes are also your pH. pH affects the structure of the protein. It can affect the bonds formed within the protein, and different pHs affect different proteins differently. So here's pH for an enzyme called pepsin that operates in our stomach. We know our stomach is a very acid environment, and so this enzyme works at a pH of 2. When that enzyme passes into the small intestine, it will be inactivated because the optimum pH of enzymes working in the intestine is going to be closer to 8. So this enzyme will not work when it moves through the digestive system. So it's a way of inactivating enzymes. We'll also we've learned about lysosomes, and lysosomes have enzymes that are digestive enzymes. Lysosomes, I'm always spelling this wrong. Lysosomes, the pH in a lysosome is also slightly lower than the rest of the cell. And so they help, it's a more acid environment, and those enzymes will not work if they happen to get spilt into the regular cell. Something else that enzymes need in order to work are cofactors. Cofactors may be non-protein enzyme helpers. They may be inorganic, such as metals, or they may be organic cofactors. If they are organic, they get a special name. They're called coenzymes. And many of the vitamins that we have in our body are actually coenzymes, and that's why it's important that you have a balanced diet and you have all the vitamins you need to help your enzymes function perfectly. So pause the video and answer some of these questions about an enzyme. What is an enzyme? Which part of the enzyme binds to the substrate? What kind of interactions can occur here at the substrate? And then 4, 5, and 6 are some nice thinking questions. What happens if you delete an amino acid on the active site region? Would it affect the overall function of the protein? What if I just swapped it for an amino acid that's very similar? Would that affect it? What happens if I deleted an amino acid in a less important region of that enzyme protein? Explain how temperature and pH affect the function of an enzyme. And how can a bacteria live at a temperature of 100 degrees Celsius when